All right, now that we've wrapped up our deep dive into heart structure, function, and control, it's time for us to explore some circulatory alternatives. So we're going to look at 4 different groups of organisms for this: fish, amphibians, and non-bird reptiles. Sorry, 3 groups really: amphibians and non-bird reptiles, and the invertebrates. And again, we're going to use bird and mammalian circulatory structure and function as sort of our starting framework for understanding and appreciating these other circulatory systems in comparison. Just a reminder, so we've seen the Hagen-Poiseuille equation as a characterization of blood flow rate, right? I also talked earlier about an alternate depiction of many of these terms with regards to vascular resistance, right? And just notice that vascular resistance is a reorganization of the parameters here in the Hagen-Poiseuille equation for flow rate. And so we can also use these to calculate flow rate in an alternate fashion as with the numerator being pressure gradient, the denominator being vascular resistance, with vascular resistance representing these other properties that influence flow rates. And I'm highlighting this because we're going to see this version of flow pop up again at some point in this lecture. So now we're ready to start exploring some alternate organizations across animals. And I want to point out that this looks really overwhelming at first when you look at it. So we're going to be stepping through this to sort of see how we go from something that looks like what we observe in birds and mammals to what we observe in other groups. And in reality, the evolutionary reality is that things happen in the opposite direction, right? So what we see in birds and mammals are more derived systems compared to simpler circulatory systems found in other animals. And again, the depictions in our textbook, there's some elements to them that are a little overly complex. So I've tried to redraw these circulatory plans to highlight some of the major features that I think you should hone in on. And I'm going to just give you the take-home message first, and then we're going to walk through the specifics. So for fish, the main thing to take away is that fish have one basic circulatory arrangement, and then we're going to see some variations on that theme based on how fish breathe. In the non-bird amphibians and reptiles, what we're going to see is that these animals have incomplete separations between parts of their circulatory system. And here's Martha to admire those circulatory alternatives as well, right? So non-bird amphibians and reptiles have incomplete separations, and even though they don't have full separations, there's still limited mixing within their hearts. And the real benefit of this arrangement is that this provides additional versatility in circulatory function that isn't present if you have no option of mixing, which is what happens in birds and mammals. Then we're going to take a look at invertebrates that have closed circulatory systems. And the big takeaway from that is that, there's no rule that says you can only have a single heart. It's possible to have more than one heart, right? If there are, physiological advantages to doing that to be able to move blood effectively. And then we're going to conclude by looking at invertebrates that have open circulatory systems. And here the message is that if your circulatory system is open, that can lead to a decrease in resistance, and that has some consequences for overall circulatory function as well. So let's get to it. So in fish, again, thinking from the circuitry standpoint, fish have a very simple circulatory circuit that just has a single pump. So I have this diagram up in the lower right of your screen, but also on this slide, just to remind us of the circuitry diagram for birds and mammals, right? So we have all of the blood passing through the systemic side of the heart, traveling through the systemic circuit, coming back to the heart, and then passing through the pulmonary circuit and being fully reoxygenated right before traveling around again. Your everyday fish has an arrangement of tissues in series as well, but blood only makes a single pass through the heart, right? So it goes from the heart to the gills, it's reoxygenated, it passes to systemic tissues, and then it returns back to the heart. And I have this question on the slide of, well, okay, what are the implications, Martha, what are the implications for the relative velocity of blood flow for mammals and fish, mammals and birds compared to fish. So think about this. Blood goes from the heart to the gills, right? At the gills, that blood needs to be reoxygenated, so it's going to pass through capillary beds in the lamellae of the gills. Then it has to come back together, travel to the systemic tissues, pass through a second microcapillary bed before it makes its way back to the heart. If you think back to the velocity of blood in birds and mammals, right, it slows at capillary beds, right, because there's this sudden large cross-sectional area, right, to traverse across, right? So if that happens at the gills, right, then there's not going to be a lot of pressure force past the gills to continue flowing that blood through the systemic tissues. So that's going to limit the rate of oxygen delivery in a fish circulatory system. Here is a view of the physical arrangement of the circulatory system in a fish. And again, we're not an anatomy class, we're a physiology course, so we're not going to be super focused on, knowing all of the intricate details of this, but it is useful to kind of see how things are laid out to appreciate the overall flow of blood. So we're going to start with the heart pumping blood to the ventral aorta, so down here at the bottom of the fish. And remember, this blood is deoxygenated, okay? So then it travels across the gills, right? The entry of blood into a tissue is called perfusion. So blood perfuses the gills, it becomes oxygenated. Then the dorsal aorta here along the spine of the fish will carry the blood to tissues, right? So this is oxygenated blood. So oxygenated blood will reach the fish's brains, the fish's eyes, digestive organs, excretory organs, etc. And then a collection of veins known as the great veins return that blood to the heart. So notice this heart is only getting deoxygenated blood after it has passed all the way through the fish's circulatory system. Okay, so this can cause some problems for fish, right? So the fish heart has to be able to generate enough pressure to get blood through the gills and through the systemic vasculature. The main pressure source in the fish's heart is also a ventricle, right, such as ours, right? But there's just a single ventricle, right, because there's just a single pump carrying out the squeezing action. Fish also have are an example of spongy myocardium, right? So the heart tissue itself is getting its oxygen supply from the blood that is passing through the heart. Okay, so if the heart consists of spongy myocardium, again, that means that it's not going to be able to generate as much contractile force, and it's getting deoxygenated blood coming back from the systemic circuit. And those seem like issues for being able to generate force effectively. This diagram is also outlining a couple of other structures in association with the ventricle. I'm not going to ask you excruciating details about these structures, but they're sort of helpful for getting oriented. So let's start with blood returning through the sinus venosus here on the right-hand side. Fish hearts do have an atrium, so analogous to the atrium we've seen in birds and mammals. Right, that's kind of a blood collection point feeding into the ventricle, but again, the ventricle is the primary squeezing unit in the fish. And then past that point, blood flows into the major coronary artery, and there's some variation here across fish species. It can be either the conus arteriosus, which can also contribute to the pumping action, or a bulbous arteriosus, which doesn't help pump. And I think we can kind of appreciate this compared to the aorta, right? The aorta itself is not generating contractile force, right? So it would be closer to the bulbous arteriosus, but it can help extend the pressure pulse from the heart. So these are problems for delivering oxygen to tissues, but it also seems like fish don't really seem to care. Well, not that they literally care or don't care, but there are still plenty of fish that can survive across a wide range of environment types. So it doesn't seem like you necessarily need to have a fancy circulatory system to function well. And fish don't really care because in general, fish have lower oxygen demands compared to something like a mammal or a bird. So just in general, they don't need as much oxygen as we do on a regular basis. So they can get away with having comparatively smaller hearts, comparatively lower cardiac output, and their circulatory system has generally lower arterial pressures, right? So at the very most, a fish heart can reach around 12 kilopascals, where we can get up to 16 kilopascals pretty easily. All that said and done, it's not as if fish circulatory properties don't matter at all, because we can see changes in heart characteristics and circulatory performance in association with the degree of physical activity that the fish engage with. So here's another another figure that we saw from our textbook. The x-axis here is the index of migratory effort for salmon reaching their spawning areas, right? So some rivers are much more intense than other rivers to spawn up. And a side note, if salmon have to travel up a more physiologically challenging river, they tend to also accumulate more body energy stores for that migration, and that tends to make those salmon more delicious than ones that have an easier migratory path. But anyway, so we have an index of migratory effort on the x-axis, and then in salmon, we actually do see some fraction of compact myocardium, right? And it's correlated with the intensity of spawning, right? So if spawning is harder, there's clearly been a selective response in circulatory function. So we do see some compact myocardium in some fish groups, right, that then gets oxygenated blood. And here again, tuna are an outlier. So tuna tend to pump their blood rapidly. They have twice as high of a weight-specific cardiac output at rest, and they have a higher blood hemoglobin concentration. So clearly there are still strong selective pressures in fish for effective oxygen delivery. So again, here's a redraw of fish circulation, summarizing kind of the major structural components, right? So just focus on deoxygenated blood returns to the heart, passes through an atrium, passes through a ventricle that contains spongy myocardium, that travels through the conus or bulbous arteriosus, to the gills, to the body tissues, and this is a reminder that we see a major pressure loss across the gills, right? So fish heart has to do all of the work of circulating blood in just a single pass through a heart. Now we get to look at some alternatives, and these alternatives show up in our air-breathing fish. Remember talking about air-breathing fish when we were looking at ventilatory structures? So in air-breathing fish, we see variations on our basic fish circulatory plan. So up here at this top picture is just ordinary fish, right? Heart, gills, tissues. What I want you to notice in air-breathing fish is that essentially the air-breathing organ circulatory path is sort of pulled out from the existing fish circulatory structure. And where that gets pulled out depends on where the air-breathing organ is located, right? So if a fish uses its mouth as its primary air-breathing organ, then blood that flows through the mouth will be oxygenated. It will mix with the deoxygenated blood returning from the systemic tissues. And so in fish using their mouth as their air-breathing organ, the heart is then going to receive a mixture of oxygenated and deoxygenated blood, which it will then pump back across the gills, back through the systemic tissues. So we've gone from a situation where the oxygen source is in series right across tissues and the heart to adding a parallel source of oxygen returning to the heart. And then just as an alternate example, for fish that use their gut as the air-breathing organ, right, in this instance, the blood passes across the gills and then across the air-breathing organ, but we have the same overall consequence of now we're seeing a mixture of oxygenated and deoxygenated blood resupplying the heart, right? So what that means is typically, even though there's more oxygen returning to the heart, there's still a fairly low oxygen saturation in blood leaving the heart. So it's an improvement, but it's not a cure-all for these fish. So that leads to kind of a bigger philosophical question. Is this alternative circuitry good or bad? It sort of depends on what you think the priority might be for the animal. Like, what matters the most? Is it most important to oxygenate the heart, or is it most important to get oxygen to systemic tissues? And to be honest, fish physiologists are kind of undecided about what's the most important. So really, the jury's out. So no strong conclusion in this instance. So then we get to another arrangement in fish, found specifically in lungfish, one of our air-breathing fish types. So here's a depiction from our textbook of this. So here's the lungfish lung. Notice that blood from the lungs goes straight back to the left side of the lungfish's atrium, right? Doesn't pass go, doesn't collect 200. And in contrast, the sinus venosus, right, which gets the systemic venous blood that's deoxygenated, the sinus venosus connects to the right side of the atrium. And in lungfish, some of the arteries don't pass through the gills, right? So we see arteries here bypassing the gills, heading straight to the systemic system. And from carrying out some functional studies of lungfish hearts, it seems that there's actually only limited mixing of the oxygenated and deoxygenated blood in the lungfish heart. So there's no physical separation, right, between the sides of a lungfish's heart. But in spite of the fact that there's no physical separation, there's only limited mixing, right? So this probably reflects just the property of laminar flow through the heart. And so we might start to wonder, okay, well, why do lungfish have this really involved organization? Well, there's a potential advantage to this. Lungfish don't use their lungs all of the time, right? If they're underwater, they're not going to be breathing air while they're underwater. So an advantage of not dividing the heart is it's then possible to strategically either flow blood through the lungs or not flow blood through the lungs, depending on how useful it is to do so. So if our lungfish is underwater, this diagram also highlights an area called the pulmonary vasomotor segment. This pulmonary artery can be vasoconstricted. If it's vasoconstricted, blood flow is going to be directed away from the lung, right? And all the energy that would have gone into flowing blood across the lung back to the heart, etc., all that energy can instead go to the circuits that deliver blood through the systemic portion of the circulatory system, right? So if our lungfish is underwater, it's not going to pass blood through the lung. Instead, it's going to have something that looks closer to the conventional fish circuitry, right? That's really useful, right? Because it takes a lot of energy to flow blood. So why flow blood through your lungs if you're not actually using them? And so here's, again, an alternate depiction of that circulatory structure, just highlighting some of the major highlights for lungfish, right? So we have limited mixing in spite of the fact that there's no physical barrier, right, between these two halves of the lungfish heart. And then we have selective flow through the lungs or bypassing the lungs, depending on whether or not the lungs are being used for breathing. Okay, that actually takes us through the fish. So up next are non-bird amphibians and reptiles. And I just want to remind you here, the theme is incomplete separations but limited mixing for added versatility. And I think we're primed to look at this now that we've seen the organization that we see in the lungfish. So to start out, I want you to think about, for amphibians and non-bird reptiles, here's your review question. How do these animals breathe, generally speaking? All right, and hopefully you have a picture in your mind. You're starting to think about breathing cycles. You're starting to think about breathing organization. And hopefully you're remembering both groups have tidal breathing structures, right? And both groups have breathing patterns that are characterized by periods of apnea, periods of non-breathing, right? Well, that is going to factor into their circulatory arrangement. So here are the diagrams, again, from our book for amphibians and for non-bird reptiles. Specifically in this case, we're looking at turtles, lizards, and snakes. A couple of things to notice. So as we saw with our frog hearts, we now see two separate atria. So we see, and this is a little backwards in these diagrams, so bear with the diagrams, right? The left side of the heart and the right side of the heart, we have a left atrium and a right atrium, right, in both of these groups, but a single ventricle, right? So a three-chambered heart overall. In the ventricle, there is an incomplete septum or incomplete dividing tissue. But even with that incomplete division, just like with the lungfish, there's only limited mixing. And what this collectively means is that it's still possible to maintain a selective distribution of blood. So, in amphibians, for example, oxygenated blood returning to the heart, right, can then be primarily delivered into the systemic tissues, and then deoxygenated blood returning from systemic tissues can primarily be redelivered back to the lungs and then also to the skin in amphibians, right, since they're doing some gas exchange across the skin. Right, so we can see that division of blood distribution with this heart structure. This also is highlighting that in some groups of reptiles, the pythons and varanid lizards, there can be a distinct ventricular pressure on either side of this single ventricle, right? So there's nothing that says it has to be uniform squeezing. So that starts to allow for lower pulmonary pressure, right? Which we saw was important for reducing the risk of pulmonary edema in birds and mammals, right? So that's that same concept showing up again in some of these reptilian groups. And then if we think about the fact that these groups engage in apnea or intermittent breathing, having an incomplete division in the ventricle is really helpful because it means that blood can be redistributed away from the lungs at times when it's useful to avoid passing, using the energy to pass blood through the lungs. So specifically, when animals are diving, right? So if they're diving, they're engaging in apnea, they're not breathing using their lungs. They might do some gas exchange across other tissues, as illustrated here, but they can save a lot of energy if they bypass the lungs and primarily just flow blood back through the systemic circuit. Right, and these guys, as with lungfish, will make some adjustments to peripheral vascular resistance, right, through selective vasodilation and vasoconstriction to redirect blood flow where it's useful. So I've redrawn these. Again, I just think it's a little simpler to have all the diagrams be consistent with the left heart at the bottom, the right heart at the top, right? But again, just highlighting how we have partial mixing in the ventricle, right, which means that it's possible to redirect blood away from the lungs when it's not useful to do gas exchange through the lungs. And this also is highlighting, although the diagram in our textbook doesn't comment too much on this, generally reptiles are not using skin as a gas exchange organ. There's some turtles that do, but they're kind of an exception. So the simplified diagram is a better depiction for most of our thinking about reptiles, non-bird reptiles. Okay, then we get to another fun and interesting exceptions in the crocodilians. And for this topic, again, focus on the point that in general, valves open or closed, right, across the circulatory system, they're one-way valves. The mechanism opening or closing them is back pressure, right, because they're one-way valves. That's the general rule for valves in circulatory systems. There is an exception in the crocodilians. So that's highlighted in this upper left diagram. For the crocodilian heart, we do see fully divided hearts. So a left and right ventricle. This is leaving out atria as a simplification, right? So fully divided hearts. But crocodilians are interesting in that they actually have two aorta on the right ventricle. Okay, so the right ventricle is the one returning blood from systemic tissues, potentially to the lungs, or alternately back into the systemic tissues. So for the right ventricle, the two aorta have two different types of valves. There's a cog valve, right? So this is a valve that can be vasoconstricted, so it operates distinctly, and then a flap valve, right, that is just responding to pressure differences. So if a crocodilian is breathing air, the cog valve will be vasodilated or open, right? And so when a crocodilian is breathing air, right, it's going to allow that blood to flow through the lungs, get reoxygenated, and then flow back through the systemic tissues. Okay, but if a crocodilian is diving, the cog valve will restrict, and in that case, blood's going to take the path of least resistance. It's going to pass through the flap valve, and so the crocodilian can bypass the lungs, right, and just direct that blood back through the systemic tissues during apnea, during diving. So that takes us through our non-bird amphibians and reptiles. So now we're ready to take a look at invertebrates that have closed circulatory systems. Again, the big takeaway is that you can have more than one heart. Right, so here we're just going to highlight the cephalopods, so that's octopi and squid. Right, and these groups have closed circulatory systems, as with the other groups we've seen so far, and they have multiple pumps. And in general, the function of a cephalopod's circulatory plan is similar to the function of fish. They maintain a high-pressure circulatory system. And so that can facilitate pretty rapid blood flow through a pretty high-resistance circuit. And I'm not going to elaborate on this too much, right? But here's this equation again, an alternate depiction for blood flow rate, right, as related to pressure difference over resistance, right? So you can get rapid flow if you have a high pressure system, if your system is also fairly high resistant, right? But then it's helpful to have more than one pump, particularly at points where you have vascular beds, right? So in this octopus illustrated here, octopi have two branchial hearts, right? These are associated with the octopi gills, right? So to flow across the gill structure. And then they have a systemic heart, which is going to take oxygenated blood from the gills and pump that oxygenated blood through the systemic circuit. So then we're going to go straight into a contrast with open circulatory systems found in aquatic invertebrates. So in contrast to what we've seen so far, in an open circulatory system, the animal's blood, which is also often called hemolymph, just to distinguish that it's not exactly the same as blood in a closed system, right? But the circulatory fluid moves directly through tissues, right? So it doesn't have to, it's not confined to capillaries. It can just travel through tissues directly. So we see this type of circulatory system, for example, in the decapod crustaceans, right? So think back to what we looked at for breathing systems for this group. And it's tempting to think, okay, it's an open circulatory system, so maybe it's kind of wimpy compared to what we see for all of the closed circulatory systems. But even though it is open, that doesn't necessarily mean it can't do anything, right? So in the decapod crustaceans, circulatory flow is still very strongly directional. So here's an example circulatory plan, right? So blood is going to pass from the heart. It's going to be pumped out along a set of major arteries. It's going to pass through the systemic tissues, right? It's going to bathe the systemic tissues, and then it's going to return back to the heart and get pumped again. And notice this is kind of a backwards arrangement compared to what we saw in the fish, right? So we're getting oxygenated blood going to the heart instead of the heart pumping and then the blood oxygenating. And again, so this is a low pressure system, right? If blood's not confined to vessels, it's just passing through tissues. It can't blow out the tissues. So it's a low pressure system. But it is not a slow circulatory system. So here's a photograph showing a procedure just to trace the path of blood. If you inject a dye and then come back sometime later, you can see where that dye travels, right? So this is showing that path of travel through vessels and then also diffusing into tissues. And so if you kind of time how long this takes, it's not a slow circulatory system, because although it is a low pressure system, passing directly through tissues also makes it low resistance, right? So if we back up to this equation, you can get fast flow rates two different ways: high pressure or low resistance. So in the absence of high pressure, we can still get fast flow through low resistance. Right, and this actually means that the decapod crustaceans can maintain much higher flow rates than fish do. So we're just going to focus. This is a very extensive comparison of circulatory properties. We don't need to know all of these excruciating details. Let's just focus on blood flow rate here. Right, so here are rainbow trout and starry flounders, so two fish species. And then contrast that with rock crabs and spiny lobsters for milliliters of blood per kilogram per minute. Much faster flow in these decapod crustaceans, right? And that's a product of, look at this, much lower resistance of those systems, right? There's not really a major difference in heart rate heart stroke volume doesn't tell the whole picture. So really it gets down to lowered resistance. All right, we're going to wrap up this topic examining tracheate arthropods, right? So recall that in the tracheate, so terrestrial arthropods, the ventilation system is decoupled from the circulatory system, right? We don't have the alternating convection diffusion pathway. We just have convection diffusion, right? The trachea directly deliver air to the tissues, and so there's just a single convection diffusion step. So there's no circulatory system required to get oxygen to tissues and to remove carbon dioxide. This is really amazing and powerful because this fact lets us ask a really interesting question about the evolution of circulatory function. And that question is, to what extent is circulatory structure and function driven by the demands of gas exchange? And to refine this question further, keep in mind that flying insects are among the most metabolically active animals in general, right? This is a distinction from crustaceans. Crustaceans don't really stand out in this regard. The flying insects, think of them as being a little bit closer to hummingbirds. They're highly metabolically active. Okay, so what are the consequences for circulatory function? I don't have it here on the slide, but I'm going to tell you, for tracheate arthropods, circulatory function is sluggish, very slow, right? So in general, the fact that the tracheate arthropods, they have very unimpressive circulatory systems. Yes, they still circulate, and actually even into wing structures, right? There are veins that will deliver nutrients into wing structures, but they're not delivering gases, right? They're just delivering nutrients. What that tells us is very clearly the primary driver of circulatory function in all the other animal groups we've looked at is delivery of oxygen. All right, that takes us through our major topics for circulatory functions, looking at the alternatives across other groups. I hope you found this to be really enlightening and interesting to ponder, to build upon what you've learned so far about circulatory function in birds and mammals. And following this topic, the last topic that we're going to explore in this portion of our course is the physiology of diving in mammals. This is a really fantastic wrap-up topic because it's going to help us synthesize what we've learned about gas exchange and circulatory function up through this point in the course. To help set the stage for this and give us some perspective, I want to compare and contrast human divers with cetacean divers, right? So some of our diving mammals. So this is a photo of a Japanese pearl diver. This individual is a member of a community that are some of the best human underwater divers globally. These individuals can spend a period of up to about 3 minutes underwater if they're not actively moving. If they are swimming, they can spend about 90 seconds underwater. So just try to visualize for yourself. You could even try to hold your breath for a minute, what that must be like. These divers can swim down to depths of about 15 to 25 meters. And now here on the right, we have dive properties for the cetaceans. On the x-axis, we've got dive duration, and y-axis is percentage of dives. So notice that cetacean divers can spend over an hour underwater during a single dive. That said, the majority of those dives are a lot shorter, but it's possible for them to stay underwater for much, much longer. And if we also examine dive depth, although most dives are fairly shallow, they can also go much, much deeper, so 600 meters compared to the 15 to 25 that a human diver could manage. So we're going to investigate what is it about the physiology of the diving mammals that allows them to stay underwater for so much longer and allows them to dive to much greater depths.